So when I was a high school student, I was absorbed in playing basketball. I didn't study so much. But I wanted to be a veterinarian. And I thought I should go to Hokkaido University. I'm here. So, Nagoya is. But I did go there because it's very far from Nagoya to Hokkaido. And another reason is here, because I found the name in some books. Names Kinji Imanishi. He's a Japanese ecologist and anthropologist. And he was the founder of Kyoto University's Prime Research Institute. And he is considered one of the founder of Japanese primatology. Because I like animals, not plants. So I want to go there. Because he graduated the same faculty of agriculture of Kyoto University. So I decided to take an entrance exam for Kyoto University. We have very different system in Japan, and we have to take an entrance exam if you want to go to any of the university. And another person is Dr. Kihara, Hitoshi Kihara. He was very famous plant geneticist. He was a professor of Kyoto University. Dr. Kihara achieved significant results in a number of studies of evolutionary biology and the genetics of higher plants, in particularly the proposal of the genome theory, which he published in 1936. His saying, such as this one, is very famous too. The history of the earth is recorded in the layer of its crust. The history of all organisms is inscribed in the chromosome. So he is a kind of a chromosome man, a chromosome scientist. Fortunately, I passed the exam. So I joined the Professor Tsunewaki's laboratory. He was the third generation professor. Just he succeeded Dr. Kihara's laboratory. So that's why I went there. And I want to learn some plant genetics. This is the first trial for me to go in the plant genetics. I'm not quite sure before I came here FYP means, but now I could understand. But we don't say FYP in Japan. Sotsuron, that means just same mean like FYP here. So I finished my FYP under Dr. Tsunewaki. The title is like this, it's very complicated. And continued to graduate school of Kyoto University. And I finished my master's thesis, the genetic study on the cytoplasm of Aegilops cylindrica host and its relatives. It's quite difficult to understand. But I could publish after finishing master's degree. After a quarter of a century, Professor Tsunewaki and I and other colleagues could determine complete DNA sequences of chloroplast and mitochondria in wheat. So, 1976, I could get an opportunity to go to study abroad. The destination is Colorado. So you could point it out, where is Colorado State? So many states in the United States. Around here, or here. So Colorado is almost in the middle. When I was a student, I should fly over from Tokyo to San Francisco, and then fly over again to Denver. The name of the university is Colorado State University. We can call it CSU. CSU founded in 1870. It was renamed in 1957. And this university has about 24,000 students, maybe same as UTAR. CSU is in Fort Collins, a mid-sized city, about 120,000 people there. And it is located in northern Colorado at the western edge of the Great Plain and also at the base of the Rocky Mountains. Here is Denver, and Fort Collins is here. And you can find Rocky Mountain National Park. So it's quite close to the national. Fort Collins is a very nice place to live. And such as Reader's Digest ranked Fort Collins as the third best city in the country to raise a family. Or an E-television featured Fort Collins as one of America's 10 most livable cities. So it was a very nice city, but I'm not quite sure about present. So CSU has 8 colleges, and they have some degree program and so on. From campus, you can see Rocky Mountains. My advisor is Dr. Takumi Tsuchiya. I'm quite sorry he passed away in 1992 due to cancer. But he was recognized for his outstanding contribution to plant genetics and cytogenetics. And he named a university a distinguished professor in 1991. I had another advisor named Dr. Eric Goose. Because my study was supported by research assistance from USDA, National Seed Storage Laboratory. And I had to study chromosomal aberration induced during seed storage. Because to get a kind of fellowship, I have to live in the United States. And I taught this laboratory to the student a couple of days ago, because this laboratory is one of the most famous seed bank in the world. It took about 3 years to complete my PhD program. And finally, I could receive a PhD, a Doctor of Philosophy degree. We don't say convocation. Instead, say commencement in the United States. So first time I came here, I was so complicated. The title of my PhD dissertation is Genetic Changes Induced by Artificial Seed Aging in Barley. From this dissertation and other related research, we could publish about 10 papers. After finishing my PhD program, I moved to Reno, Nevada. But this is the first my postdoctoral experience. But I didn't like Reno, Nevada so much because it's a kind of gambling city. So you can find a lot of casinos and so on. So not good for a student, I think. My supervisor was named Dr. Baldev B. He is from India originally. And he taught me how to prepare human chromosome, so not plant. And I met Dr. Michael and Comfort when he was young. Maybe he was a master course student. But now he's a professor of University of Texas. So I didn't like Reno so much. So I want to move some area. And then in addition, I want to learn plant tissue culture technique too. So I decided to move to Davis, California. So UC Davis is founded 1908 as the university farm. And Davis is the largest of the 9 UC campuses in area, and second largest in budget, and third largest in enrollment. Davis is a very nice place to study and to live, because not so many cars on campus, because almost students use bicycle. My supervisor was Dr. Tom Orton. He was as old as me. He just finished his PhD program, but he could get an assistantship at Davis. So he was a quite smart guy. And I also see Steve Huxley. He's also famous for plant breeding and plant genetics in tomato. He was the last student of Dr. Charles Lake. He was also famous for tomato genetics at UC Davis. I met Dr. Chao Chen Jian. He's a sunflower geneticist at Fargo. I learned at Davis plant tissue culture technique as well as molecular biological techniques. At Davis, I analyzed chromosomal and somatochronal variation in vitro, meaning in tissue culture, and also revealed that the C genome of brassicas, such as Brassica oleracea, has very high regeneration capability. And I also developed new chromosome preparation technique using protoplast, that's kind of naked cell of plants. If you can treat enzyme, you can take out the cell wall. And those protoplasts, their characters are almost same as animals. So you can apply animal chromosome preparation technique to plant protoplast. And then we could get a very nice chromosome image. And I came back to Japan because I could obtain the position in Nihon University. It was also a nice place, but not good for research work because we had very heavy teaching duty. But I just tried to publish one paper here. And that is effect of auxin and cytokinin on the induction of sister chromatin exchanges in culture cell. You may know sister chromatin exchanges don't affect any genetic information because both chromatin has same DNA information. Even if exchange occur, no genetic changes there. But you can count some sister chromatin exchange numbers as correlated with mutagenetic effect. If you add some strong mutagen, you can find a lot of SCEs in the cell. And then I found the herbicide 2,4,5-T is a strong, very strong mutagenic effect. It has